Okay, so today's topic is microtubule-based transport, which is of course important for the intracellular organization and cell function. And it's particularly critical in highly polarized cells, such as neurons, in which material has to be moved over vast distances. Here we have an illustration of axonal transport, which occurs along microtubules which are uniformly polarized, such that the plus ends face the growth cone or the synapse, and the minus ends face the cell body. And on the right, I've listed some of the cargo that is transported in axons. These can be small vesicles, organelles, RNP granules, or protein assemblies, such as neurofilaments. And one example is, of course, the synaptic vesicle components that have to be transported from the cell body to the synapse. This would be through anterograde transport. And on the other hand, components have to be transported from the periphery to the cell body to be degraded or recycled. And this occurs through retrograde transport of endo-lysosomal organelles. And there is two types of motors that transport all this diverse cargo, called kinesin and dynein. Kinesin walks towards the plus end of microtubules, so the kinesins are responsible for anterograde transport in the axon, and dynein walks towards the minus end of microtubules, so it is responsible for the retrograde transport. And in most cases, both types of motors are present on the cargo. So although cargo has some bias in terms of the overall movement, essentially all cargos can move in both the anterograde and anterograde direction. So a major unsolved question is how these two types of motor coordinate with each other so that they are setting up a useful bidirectional transport system. Now, the importance of axonal transport is illustrated by the fact that axonal transport defects are hallmark of neurological diseases, including very prevalent age-related disorders such as Alzheimer's and Parkinson's. And as summarized nicely in this review by Lula Vandenborst's group, it is now clear that axonal transport defects are both caused by and contribute to the pathological pathways that are implicated in neurodegenerative disorders. There are examples where mutations in genes that encode for axonal transport components cause disease. For example, mutations in the dynein cofactor dynactin cause motor neuron disease and Parkinsonism. And so in these instances, the relationship between axonal transport defects and neurodegeneration might be quite direct. And it's from a medical perspective, if one understands the mechanism of how axonal transport is regulated, then maybe eventually one could intervene pharmaceutically to boost transport in these diseases, and this may help to slow down disease progression. Now, a key step of regulation takes place at the interface between the organelles, or between the cargo and the motors, and this involves so-called adapter proteins, which recruit the motors to the cargo. These adapters are, in the case of vesicles and organelles, are effectors of Rab small GTPases. So they bind to the GTP bound active form of the Rab. And once they're bound to the Rab on the organelle or vesicle, the adapters then recruit the motors to the cargo. Now, there are about 40 kinesins in vertebrates, and pictured here is the conventional kinesin called kinesin 1, which I'm going to talk about. But there's only one dynein And so while there's some specialization of the different kinesins with respect to the cargo that they transport, the dynein is sort of the all-purpose -end-directed motor. And so dynein really relies on adapter proteins. It has to interact with adapters to be able to selectively bind to cargo. Some adapters are exclusive for kinesin, some are exclusive for dynein, and then there's a category of adapters that bind to both motors. And this implies that these types of adapters may be able to regulate transport in a bidirectional manner. However, these adapters remain in general poorly characterized. And so today I'm going to talk about one of these adapters that can bind both to kinesin and dynein. And this adapter is called JIP3. JIP3 stands for junk interacting protein 3. It's a MAP kinase scaffold, and its role in axonal transport was first described about 20 years ago in Larry Goldstein's lab for the Drosophila homolog Sunday driver. And since then, a number of labs have shown that this function of JIP3 is broadly conserved in invertebrates and vertebrates. Now, the main signature defect of JIP3 inhibition is that lysosomes become highly abundant in the axon. And this is shown here in these mouse neurons. Usually the axons do not have many lysosomes, but in this JIP3 knockout, you can see that there's a massive accumulation of lysosomes in the axon. And in invertebrates, this is also the case for other organelles, such as endosomes and the Golgi. So genetically speaking, JIP3 acts as a negative regulator of axonal organelle abundance. But it's not really clear how this phenotype rises. And one hypothesis is that the interaction between JIP3 and dynein is what is required to clear lysosomes and other organelles out of the axon into the cell body. And another interesting question is whether this JIP3 kinesin 1 interaction participates in organelle transport, because so far there's only evidence that the JIP3 kinesin 1 interaction participates in the transport of small vesicles. So my lab has been interested in understanding the function of this JIP3 and motor interactions. And one way we thought we would approach this is to engineer and then characterize specific mutations that displace either only dynein or only kinesin from JIP3. And so for this, we need to understand, of course, how the two motors bind to this JIP3 protein. Here's a schematic of the domain architecture of JIP3, and we know already quite a bit about how kinesin binds to JIP3. There's two binding sites. One is at the end terminus of JIP3 and involves this RILP homology RH1 domain. And this is the binding site for kinesin heavy chain, and more specifically for the kinesin heavy chain tail domain. And there's a second binding site for kinesin light chain, and this binding site is in this leucine zipper domain. By contrast, it wasn't really known how JIP3 interacts with dynein, but we knew that it involved one specific subunit of dynein called dynein light intermediate chain, which is diagrammed here. So dynein is a megadalton complex, and dynein light intermediate chain is one of the subunits. It consists of an N-terminal RAS-like domain, and then it has a flexible C-terminal tail. And in this tail, there is a short helix, helix 1, and we and others have shown that this short helix binds to the N-terminus of various dynein adapters, which contain one of three folds, either a coiled coil motif called the CC1 box, a hook domain, or an EF hand. But JIP3 doesn't contain any of these folds. So we started by asking whether and how this helix 1 of dynein light intermediate chain interacts with JIP3. And we used purified recombinant human proteins to do these binding assays. And we did it for JIP3 and its paralog JIP4, but also for the RILP family proteins, which in vertebrates consist of RILP, RILPL2, and RILPL2. RILP is an adapter of dynein for lysosomes, and RILPL1 and RILPL2 participate in celiogenesis. And so we did these in vitro binding assays, and we found that this helix 1 was required and sufficient to bind to the N-terminal RILP homology, this RH1 domain. And this was the case for JIP3, JIP4, RILP, and RILPL1. We did not find any interaction with RILPL2, which makes sense because this is an adapter for the actin-based motor myosin Va. And so we wanted then to understand more precisely how this interaction occurs, and we got some clues from structural work which had been done on the interaction between myosin Va and RILPL2. So this interaction involves a hydrophobic pocket, which is formed by a 4-helix bundle in the RH1 domain. So we have here a prediction of the structure of the RH1 domain of JIP3. This is by AlphaFold 2. And this hydrophobic pocket that I'm talking about is formed by the alpha-2 helix and the N-terminus of the alpha-3 helix. And so you have 2 hydrophobic pockets per RH1 domain. And in this publication, it was found that there's a helix of myosin Va that inserts into the hydrophobic pocket of RILPL2. And when this conserved valine here was mutated to glutamine, this was sufficient to displace the myosin Va helix from this hydrophobic pocket. And so we thought maybe the helix 1 of dynein light intermediate chain uses the same hydrophobic pocket on JIP3. And in fact, when we asked AlphaFold 2, where would you put this helix 1 of dynein light intermediate chain, it puts it exactly where we think it would be, which is into this hydrophobic pocket. So here's the helix of dynein light intermediate chain. One of the residues we're going to mutate is highlighted here, phenylalanine. And here's the critical valine in the hydrophobic pocket of JIP3 that we're going to mutate. So to test if this helix 1 of dynein light intermediate chain really uses this JIP3 hydrophobic pocket, we mutated these 2 residues. We performed ITC with purified proteins. And so we used the C-terminal tail of dynein light intermediate chain and the RH1 domain of JIP3. You can see that the wild-type proteins bind each other with a dissociation constant of about 1 micromolar. This is similar to what has been observed for the interaction between dynein light intermediate chain and other dynein adapters. If we then mutate residues in the light intermediate chain helix 1, we lose this interaction. And if we mutate the RH1 domain, if we make this valine V60Q mutation, we also lose the interaction. So we have now a point mutation in the JIP3 RH1 domain that prevents the binding of dynein light intermediate chain. Now I'm coming back to this slide because the RH1 domain is of course also the interaction site for kinesin heavy chain. And so we next asked whether our point mutation also affects the interaction between JIP3 and kinesin heavy chain. And we address this by gel filtration experiments that you can see here. We combined the JIP3 RH1 domain with the C-terminal tail of kinesin heavy chain. And when we do that, we see a shift in the elution profile. So essentially the RH1 domain now partially coelutes with the kinesin heavy chain fragment. You can see this also in the solution profile. And what's notable is that we get more kinesin heavy chain out of the column when we add the RH1 domain, which, so if you compare this elution with this elution, there's more kinesin heavy chain here when the RH1 domain is present. And this indicates that the binding between the kinesin heavy chain and the RH1 domain of JIP3 has a stabilizing effect on this kinesin heavy chain tail fragment. And we see exactly the same effect if we now use the RH1 domain in which this valence 16 is mutated to glutamine. And so from these experiments, we can conclude that the JIP3 V62Q mutation does not affect the interaction between the JIP3 RH1 domain and the kinesin heavy chain C-terminal tail. And so the conclusion then would be that this JIP3 V62Q mutation is a separation of function mutation. And so this would be a useful tool to investigate the dynein-dependent functions of JIP3. Now, to examine the effect of this mutation in vivo, we turned to our model organism C. elegans, where we can use live imaging and we can easily do genome editing. And the JIP3 homologue is called UNC-16 in C. elegans. We introduced this point mutation into UNC-16, which is V720Q, and we compared it to two null mutations. One has a premature stop codon, and in the other case, we've deleted the entire UNC-16 locus. And you can see on this Western blot that the two null mutations don't express any UNC-16, whereas the UNC-16 V720Q mutation is expressed at the level that is similar to the wild type. And to determine whether this mutation is important for UNC-16 function, we looked at animal locomotion. So we assessed the frequency of body bends of these nematodes in a liquid droplet. And it's known that if you knock out JIP3, these animals are almost paralyzed, as you can see here. And it turns out that the point mutation that we introduced has the same effect. And so this shows that the interaction between UNC-16 and dynein light intermediate chain is important for UNC-16 function. Now, we next examined the distribution of organelles in touch receptor neurons, which make these long processes that are directed towards the nose of the worm. And here is one of these neurons called ALM. And so we used markers for different organelles. We started by looking at lysosomes using CTNS1 MK2. And you can see in the control, most of the signal is concentrated in the cell body. There's almost no signal in the actual neurite. There's a few lysosomes here at the beginning of the neurite. And if we look at the knockout of UNC-16, lysosomes are now distributed all along the axon and along the neurite, which is what has been described previously and is consistent with what is observed in vertebrate neurons. The UNC-16 point mutation also has less signal in the cell body, but the distribution in the neurite is different. The signal is now primarily concentrated at the neurite tip. And we can visualize this by plotting the fluorescence profile along the neurite from just after the cell body, which would be 0% here, to the neurite tip, which is 100%. And so you can see, in contrast to the null mutation, the point mutation is causing this accumulation of signal at the neurite tip. And to illustrate this further, we can zoom in to the cell body or the neurite tip. You can see in both the knockout and the point mutation, the cell body signal is decreased, but only the point mutation results in this increase of signal at the neurite tip. And this is also obvious here from the quantifications of the signal. And this is not just the case for lysosomes. We also looked at early endosomes, so RAP5 as a marker, late endosomes, RAP7 as a marker, and 6-7, which labels early recycling endosomes. And in all cases, we saw that in the point mutation, you have a massive accumulation of signal at the neurite tip. The most striking example is RAP5. In this case, the point mutation is almost a mirror image of the control. So most of the signal is now at the tip. There's almost nothing left in the cell body. And again, you can see this by plotting the fluorescence profile along the Neurite. I had to split up the y-axis here for you to see that in the knockout, you do have a general increase in signal in the Neurites, but that is dwarfed by the accumulation at the Neurite tip in the point mutation. And again, you can see that when you zoom in to the cell body or the Neurite tip. To understand why we have this massive accumulation at the Neurite tip, we looked at the motility of these RAP5 particles, and we used chromographs to analyze how these particles move. In the control, you can see that they move in a bidirectional manner. In the knockout, they're mostly stationary. And in the point mutation, the effect is distinct from the knockout in that you have less particles that move in the retrograde direction, but the anterograde moving particles are similar to controls. So we have basically perturbed retrograde transport of these particles, but we have not perturbed anterograde transport of these RAP5 particles. And so that offers an explanation for why these particles then accumulate at the Neurite tip in this UNC-16 V720 point mutation. And so overall, this suggests that the interaction between UNC-16 and dynein-light intermediate chain is essential for the retrograde transport of endo-lysosomal organelles. Now we wanted to understand which are the kinesins that are responsible for the Neurite tip accumulation of the endo-lysosomal organelles in this point mutation. And so a good candidate, of course, was kinesin 1, because we know that kinesin 1 directly binds to UNC-16, and we know that we have not perturbed the interaction between UNC-16 and kinesin 1 in this V72Q mutation. And so what we did is we used a partial loss of function allele of kinesin heavy chain, which is in C. elegans called UNC-116. And so when we combine the point mutation in UNC-16 with the mutation in the kinesin heavy chain, we now see that we lose the Neurite tip accumulation for both the lysosomal marker and the early endosome marker. And this is accompanied by an increase in the signal in the cell body, as you can see here. So from this, we conclude that kinesin 1 activity is required for this Neurite tip accumulation of endo-lysosomal organelles in the UNC-16 V720Q mutation. But the question now was, is it really the kinesin 1 that is bound to UNC-16 that is responsible? And for that, we were looking for a mutation that would selectively displace kinesin 1 from UNC-16. And we settled on this mutation here, which in humans is L444P. This is a mutation in a kinesin light chain binding site. And it was described a couple of years ago in a paper that showed that this mutation causes intellectual disability with brain anomalies. So this is a disease mutation. And in this paper, they performed computational modeling to show that this mutation most likely perturbs the binding between CHIP3 and kinesin light chain. And so to confirm this, we used purified recombinant proteins, and we introduced this point mutation into the mouse CHIP3 protein, and so in this case, it's L439P. And so we asked, can a CHIP3 fragment that contains this kinesin light chain binding site still bind to kinesin light chain when this mutation is present? So we performed pull-down experiments in vitro, here you have the input, you have the wild type CHIP3 fragment and the mutant CHIP3 fragment. This is the kinesin light chain. If we do the GST pull down, we see that the kinesin light chain pulls down the wild type CHIP3 fragment, but it does not pull down the CHIP3 fragment that contains this L2P mutation. And so this confirms that this disease mutation does prevent interaction between CHIP3 and kinesin light chain. And so we went back to C. elegans with this information and introduced this L2P mutation into our worms. And we then combined the point mutation in the RH1 domain that displaces dynein light intermediate chain and the mutation in the kinesin light chain binding site. And when we do this, we, as you can see here, we no longer observe this accumulation at the Neurite tip in the double mutant. And if we look at the signal at the Neurite tip, it is now clear that the double mutant is similar to the knockout and is distinct from both of the single mutants. So let me summarize the effects we saw with our CHIP3 mutants. In the wild type case, CHIP3 or UNC-16 binds dynein light intermediate chain, kinesin heavy chain, and kinesin light chain. And in this case, when we look at endosomes and lysosomes, most of the signal is concentrated in the cell body. When we knock out CHIP3, we see a shift of the signal into the neurite. This is exacerbated when we just displace dynein light intermediate chain from CHIP3. In this case, the signal concentrates at the neurite's tip. And we can rescue this tip accumulation by additionally mutating the binding site for kinesin light chain. So in this case, the distribution again resembles that of the knockout. So we can take two conclusions from this. One is that the CHIP3 dynein light intermediate chain interaction is important for the retrograde transport of endosomes and lysosomes. And the other conclusion is that the CHIP3 kinesin light chain interaction is able to drive the anterograde transport of endosomes and lysosomes. And so as a conclusion, we think that both CHIP-bound dynein and CHIP3-bound kinesin 1 contribute to the transport of endosomes and lysosomes in axons. So CHIP3 really acts as one of those scaffolds that can drive the bidirectional transport of organelles. And it's already known that CHIP3 can activate kinesin in vitro, so that provides a rationale for how CHIP3 can drive the anterograde transport. It's a bit less clear how CHIP3 drives the retrograde transport via dynein, because CHIP3 lacks some of the features that are usually associated with dynein adapters that are able to activate dynein motility. So in the future, it will be interesting to see whether CHIP3 can really activate dynein on its own, or whether it needs to collaborate with another adapter to do this. So with this, I would like to acknowledge the 4 postdocs who've worked on this project: Daniel, Bernardo, Ricardo, and Artur, and also our collaborators. And I thank you very much for your attention.